Hi, I'm Saranya. So over the summer, I did research in Colombia on the factors affecting membrane protein expression. Proteins that are generally found in the cytoplasm, like actin, have a globular shape with a hydrophilic exterior and are called cytoplasmic proteins. On the other hand, proteins that are found in the cell membrane, here, are hydrophobic and are called membrane proteins. Secreted proteins have signal peptides that are cleaved off after they cross the membrane. We can understand how a protein works by knowing its structure. X-ray crystallography is the main way to determine the structure of a protein. One way to make a lot of proteins is to use a recombinant protein expression system. In this system, a plasmid is cloned to code for the protein of interest and is transfected into a bacteria. The bacteria is grown in a culture to a large volume, and the protein is harvested from the bacterial culture. We know the structures of nearly 17,000 cytoplasmic proteins, but only of about 200 membrane proteins, even though 30% of all proteins are membrane proteins. This is because membrane proteins are harder to express and purify using recombinant expression systems. My lab has been trying to figure out ways in which we can improve membrane protein expression levels. The study in our lab showed that whenever rare codons are used, protein expression is lower. This indicated to us that factors at the transcription level in the nucleus could also play a role in protein expression. Because mRNA plays a major role in protein expression, we hypothesized that mRNA folding plays an important role in regulating protein expression levels. We decided to test our hypothesis by testing whether there was a correlation between mRNA folding energy and protein expression levels in the E. coli genome, because E. coli, a bacteria, is commonly used to express various proteins in large quantities. The lab that I worked at is part of the Northeastern Structural Genomics Consortium, which has a database of the expression levels of E. coli proteins. First, I categorized these 4,146 proteins using TMHMM, which distinguishes between transmembrane and soluble proteins. This program uses a hidden Markov model to classify proteins into transmembrane, which is here, and soluble proteins based on a number of factors, including hydrophobicity of amino acid residues, charge bias, and helix length. The first picture represents a cytoplasmic protein because it is highly probable that the peptide is present in the cytoplasm throughout the entire sequence. However, in the second picture, we found a transmembrane protein where parts of the sequence are here, pass through the membrane. There were around 3,000 cytoplasmic proteins and 650 transmembrane proteins. Then I used two programs, Signal P and LipoP, to identify proteins that contain signal sequences. This is the typical output of Signal P. The first figure predicts that the protein will be a signal protein. The second figure has no significant C-score or S-score, so it is classified as cytoplasmic. There were 450 signal P proteins and 100 signal P2 proteins. Signal P and signal P2 are signal proteins and lipoproteins, respectively. We then sent the complete E. coli genome sequence to a lab at Williams College, where they performed mRNA folding energy calculations. This program divided all the E. coli genes into 10 windows, each with around 48 codons, and performed mRNA folding energy calculations. Then I wrote computer programs to assign the correct energy values to the four major classes of proteins that I had previously classified: cytoplasmic, transmembrane, signal P, and signal P2. In order to see whether there was any real difference in the folding energies of these various classes of proteins, I wrote another program to divide the proteins based on folding energy intervals. Then I made a histogram of the percentage of proteins that occupied different energy levels in all four classes of proteins. So these are the different four classes of proteins. This is the energy levels, and this is the percentage of proteins for each class. These results suggested to us that there is a clear difference in the percentage of cytoplasmic and transmembrane proteins at each energy level. We then performed logistic regressions on the mean free energies to determine whether our data was statistically significant. Since the chi-score values are low, we know that our results are significant. Previous studies have suggested that only the first 50 nucleotides are important in terms of regulating protein expression. Our results show a significant difference between transmembrane and cytoplasmic proteins in this region. There is no significant difference in the RNA folding energies of transmembrane and cytoplasmic proteins in other windows. From this research, we learned that there is a significant difference in the mRNA folding energies of transmembrane and cytoplasmic proteins in window 1, which contains the first 51 codons that regulate protein expression. We also learned that having a strong folding energy might increase translation efficiency by preventing mRNA aggregation. This aggregation clumps the mRNA together and makes it difficult to translate, as you can see here. So in this picture, it won't be able to translate into a protein because it's aggregated together. But here, since there's a greater folding energy, it's able to translate into a protein. Finally, we learned that mRNA folding energy calculations can be effectively used to predict protein expression levels and can aid in the design of novel mutations to enhance expression levels of transmembrane proteins. In the future, I hope to continue research on protein expression levels and folding energies. We are planning on performing synonymous codon experiments to verify our results. If we change different codons in a protein sequence to codons that code for the same amino acid, we can see if this changes the protein folding energies. Another study that we can do is one on untranslated regions. mRNA may be regulated by nucleotide sequences that occur ahead of the start codon. This is a UTR, the untranslated region right before the start codon. We are planning on examining whether the folding energies of untranslated regions regulate protein expression. Okay, thank you.